Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, titled Solutions for Clinical Grade Manufacturing of Gene-Engineered NK Cells. I'm Casey Nevins, an editor at BioInsights, and joining me today is Maria Delso Vallejo, who will address how to efficiently manufacture gene-engineered NK cells for clinical applications using an automated and closed system. After the presentation, we will have a live Q&A session. We invite our audience to pose their questions to our speaker using the Ask a Question box at the bottom of your screen. We'll try to get to it during the session. I'd also like to draw your attention to the Resources tab on the right, where you can find more information on the topics covered today. Now, I'd like to introduce our speaker. Maria is responsible for the clinical NK cell applications portfolio at Miltenyi Biotec. She received her PhD in biology at Hanover Medical School in 2018. While studying for her doctorate, Maria worked as a Marie Curie early-stage researcher, studying the ex vivo expansion of NK cells for their use in immunotherapy against cancer. In her current role, Maria works closely with R&D to advance the development of tools to enable NK cell therapy manufacturing. Without further ado, I'll hand you over to Maria to kick us off with the presentation.
Hello everyone, and welcome also from my side to this webinar. Before we get started, this is a disclaimer on the Miltenyi Biotec products that will be mentioned during the webinar. First, we are going to start with a short introduction to current insights on the NK cell therapy landscape to move on to the main part of the webinar, which is about the manufacturing solutions for gene-engineered NK cells. Next, we will learn how we can enable the robust ex vivo expansion of NK cells and flow cytometry solutions for the iPSC-QC in NK cell therapy manufacturing, and finalize with a Q&A session. Let's begin with a brief refresher on NK cell biology. NK cells are cytotoxic lymphocytes of the innate immune system, characterized by the expression of the CD56 molecule on their cell surface and the absence of CD3 molecule. They are primarily present in peripheral blood, but also they are present in a certain level in some other tissues, and their main functions consist on the elimination of virally infected and malignant cells by direct recognition without any antigen priming needed. They are able to do so in an MHC unrestricted manner by recognizing their targets through a complex array of activating and inhibitory receptors. Why then NK cells are used in immunotherapy and in particular for the treatment of cancers? Due to their biological features, adoptive transfer of NK cells has been proved so far to be of low risk of graft versus host disease, as well as of cytokine release syndrome or neurotoxicities. NK cells are perfect as cell therapy in the allogenic setting, as can be generated from multiple donors as well as different cell sources. And therefore, they can be used to treat multiple patients and ultimately generate off-the-shelf cell therapies. They are potent effector cells with different anti-tumor activities. First of all, they can display their inherent ability to recognize malignant cells with their innate activating receptors. This is the so-called natural cytotoxicity. But of course, they can also be engineered to express chimeric antigen receptors, for instance, CARs, to display CAR-mediated cytotoxic responses. And also, because they express the Fc receptors on their cell surface, they can also display antibody-dependent cell-mediated cytotoxicity, or shortly ADCC. Therefore, they are a perfect fit for the combination with monoclonal antibodies or other cell engagers. There are also certain limitations to keep in mind, such as their limited persistence in the body. So in cases they need cytokine supplementation after adoptive transfer, and when used in the allogenic setting, they could be potentially rejected by the recipient's immune cells. However, there are several strategies being designed to overcome these limitations. So overall, NK cells represent an attractive cell type for immunotherapy, and this is reflected in the increase in the number of clinical trials for NK cell-based therapies, in particular since 2016. Still, most of these trials are on phase 1 and 2, but it is expected to observe a progression of these trials to further phases in the upcoming years. The NK cell therapy field is a blooming field, and we see there is even more to come as there are still many pipelines on preclinical development. Probably many of you are working on these pipelines at this time, and in particular, they are used or based on the use of engineered NK cells, such, for instance, the generation of CAR NK cells. Therefore, there is the need to establish manufacturing tools for engineered NK cells. But before we discuss about that, I would like to take a moment to make a quick remark. The journey to develop any cell and gene therapy is long and involves different stages until finally a specific therapy gets approved if everything goes well. During this journey, the manufacturing workflow as well as the analytical assays, they are getting defined and also progress according to the phases as shown in this simplified schematic. So the more the therapy progresses into further clinical phases, the more fixed the manufacturing and also the analytical assays get, and there is less flexibility and room for change. So my point here is about the importance of thinking translational during the preclinical stage and evaluate whether the research workflow that we have been following so far, whether it is easily translated into a GMP compliant clinical grade workflow or whether we need to apply certain changes to make it as a GMP compliant workflow. From Miltenyi Biotec, we are committed to provide tools to enable the smooth translation of your preclinical workflow into clinical grade manufacturing. Here, I just wanted to quickly show you our translational workflow solutions for engineered NK cells. And I will not go into detail, but basically the products in the upper part in purple correspond to research use only products that can be used throughout the different steps of the workflow. And in the lower part in blue are shown the corresponding GMP compliant version of the same products. And in green are shown different flow cytometry tools for NK cell analytics. After this short introduction, we are now ready to dive into the manufacturing of gene-engineered NK cells for clinical applications. The manufacturing process of gene-engineered NK cells, similarly to other cell therapies, involves many different steps, as we can see in this representative workflow to manufacture CAR NK cells. All these steps, when performed in an open way, using different devices and manual handling, lead to more window for risk and failures due to this higher level of manual handling. But in addition, all these open steps require higher cleanroom requirements, more highly skilled personnel, as well as multiple service contracts for all the different instruments needed, just to name a few handicaps that altogether increase the operational costs. However, we can overcome all these limitations by integrating all these individual open steps into a single closed system, such as it is the CliniMACS Prodigy platform that probably some of you are already familiar with, and the CliniMACS electroporator. The CliniMACS Prodigy platform helps to simplify the complexity of your cell processing through the different stages of your cell therapy development. It is an integrated and fully automated platform where all steps are performed in a closed fashion to ensure an aseptic process. Samples can be taken at different stages of the manufacturing process for in-process control and quality control. The integrated software has a high level of flexibility, and the user can modify multiple parameters. Ultimately, the platform has been clinically validated by different regulatory bodies, and in addition, the CliniMACS Prodigy combined with the CliniMACS electroporator enables maximum flexibility for the efficient electroporation of different cell types. Here we can see a close-up view of both instruments with some important parts highlighted. For instance, we have the peristaltic pump that enables the flow of both cells and reagents through the platform, in combination with a tubing set, which is a single-use disposable. The touch screen that allows the easy control of the processes and editing of certain parameters, the magnet unit for separation of cells, or the centrical unit that houses the chamber attached to the tubing set and enables multiple functions, such as, for instance, the sample preparation, to do washings of our starting material, or the culture of our cells. On the bottom part, we can see the CliniMACS formulation unit that is the last addition to the platform in order to automate the final formulation of the cell product. And in regards of the CliniMACS electroporator, we can see closely the electroporation cuvette and the tubing with individual bags to keep the cells and nucleic acids separated before electroporation. Coming back to our main topic for this webinar, we are focusing on the manufacturing of gene-engineered NK cells. And as you probably know, there are two main methods that can be applied for the genetic modification of NK cells. On one hand, we have transduction using viral vectors, and on the other hand, transfection of nucleic acids or similar molecules via electroporation. First, let's have a look at the use of viral transduction to genetically modify NK cells. We have just seen this workflow that summarizes the steps to manufacture gene-engineered NK cells and got familiar with the CliniMACS Prodigy platform. Using this platform, we have now developed the CliniMACS Prodigy NK cell transduction system to enable the closed and automated manufacturing of engineered NK cells via viral transduction. The CliniMACS Prodigy NK cell transduction is actually the combination of two processes. First, the CliniMACS Prodigy LP356 system is used to deplete CD3 positive cells with one tubing set, and then the pre-depleted cell fraction is transferred to a second tubing set and use the CliniMACS Prodigy PD56 engineering system to proceed with the enrichment of NK cells based on CD56 expression and continue with the activation, transduction, and expansion of the cells to finally harvest the cell product. For every step of the process, we provide all the reagents and consumables necessary, as well as dedicated flow cytometry solutions to facilitate the IPCQC of the manufacturing process that we will see later on. In the upcoming slides, I would like to show you some exemplary data on the performance of the CliniMACS Prodigy NK cell transduction. In this first slide, we can see the performance data after the separation process, after C3 depletion and 56 enrichment. And as you can see, the NK cells after this step, they are highly viable and we get very high purities with low contaminations of T cells and B cells, as we see by the low p-values in the second figure, but also low presence of monocytes. Overall, after separation, we have around a 40% recovery of NK cells. After separation, if we want to apply viral transduction to engineer our cells, it's when we stimulate or activate our cells and then transduce. So this step is flexible. You can define your parameters. And here I just wanted to show you the parameters that we have defined based on the particular vector that we are using. We are using, in our case, a baboon envelope pseudotype lentiviral vector. And in order to get good transduction efficiencies, we followed this protocol. So first of all, right after separation, on the first day, we stimulate the cells with a cocktail of cytokines. On the second day is when we introduce the vector together with the MACS GMP vector fusing 1 in order to enhance the transduction efficiency, and we apply 2 hours of spinoculation. And finally, on the third day, we washed out the vector and the cytokines, and we start the cultivation of the cells. And as you see, with this protocol that we have defined, we get nice transduction efficiencies. So by day 14, around more than 60% of the cells on average have been transduced, and they stably express the construct that we integrated. Now in this other slide, you can see how nicely the NK cells are able to expand in the CliniMACS Prodigy chamber after activation and transduction. So in our protocol, we expand the cells for up to 14 days, but you can define the duration of your particular process. And you can see here we still have some donor-to-donor variations, but overall we get nice expansions and high cell numbers of the final NK cell product. Nevertheless, we are aware that perhaps these numbers are still not enough depending on the type of application that we want to use the cells for. So we are really aware of that, and we are already working on developing a new process to enable larger scale expansion of NK cells. This is what we are calling NK cell transduction large scale, and our goal is to generate on average around 10 to the 10th CAR NK cells. So overall, the final cell product, again, when we check purities and viabilities, all the values are very high. Almost 100% of the cells are NK cells with around almost 90% viability. Really almost no contamination of T cells, B cells, or monocytes, and we get nice cell counts after 2 weeks of expansion. But not only viability and high yields are important, we also check that indeed the cells that we engineered and expanded, they were functional. And this is an example of one of the runs that we did. In this case, we tested the CAR-specific mediated killing of our generated NK cells, CAR NK cells, and we basically incubated them against a tumor cell line that was rather resistant to natural cytotoxicity with or without the expression of the target of the CAR that we integrated. And as you see, our CAR NK cells, they were nicely able to specifically kill the target cells that were expressing the target. And not only that, they were also able to release a nice profile of cytokines. And then when we correlated the levels of the cytokines, in particular GM-CSF, to the levels produced by CAR T cells, we observed that the profile or the level of this cytokine produced was much lower compared to T cells. So again, reflecting what has been observed and regarding low risk of cytokine release syndrome. Of course, this is in vitro data, but it has been already shown in a clinical setting. So overall, to summarize the CliniMACS Prodigy NK cell transduction, this process is a complete automated process that enables separation, transduction, and NK cell expansion in a closed system. We provide all the consumables necessary for this process, and when using this process, it's possible to get robust and reproducible manufacturing of virally engineered NK cells. The process also enables to just perform separation and expansion of the cells. So transduction is actually an optional step, and we also provide technical support and the training needed in order to master this process. Besides applying viral transduction to engineer our NK cells, we can also be interested in applying genetic modifications using nucleic acids and electroporation. We can perform complex genetic engineering of NK cells using the CliniMACS Prodigy platform and the CliniMACS electroporator. So far, we have optimized parameters for this small-scale electroporation of NK cells by using the test cuvette adapter provided with the CliniMACS electroporator. And with these optimized parameters, we have generated some data for proof of concept using mRNA coding for the GFP protein. As you see here, using these optimized parameters, we can reach high electroporation efficiencies, in this case around 85% of the cells were expressing GFP after electroporation. But most importantly, the viability of the NK cells after electroporation was over 90% and comparable to that of non-electroporated NK cells. And after electroporation, the GFP expression was very high for approximately 5 days, and afterwards started to decline progressively as expected, because it's not an integrated genetic modification. Furthermore, we also looked at the expansion of the cells and their functionality. So the electroporated NK cells expanded in a similar way to non-electroporated NK cells and displayed a comparable cytotoxic response against a particular target cell line, as well as cytokine production. If we need to perform complex genetic engineering of our NK cells, it can be difficult to translate such a process into a clinical grade manufacturing process. But the CliniMACS Prodigy and the CliniMACS electroporator can facilitate the performance of such complex steps, and therefore, if you are interested and looking into this, I invite you to consult with us to discuss your specific needs. To wrap up this chapter, I just would like to have a few words on upscaling options for NK cell manufacturing. The CliniMACS Prodigy and the electroporator, we have just learned that they can enable to automate the key steps of our complex manufacturing process, such as the separation, engineering, and the preliminary expansion of the cells. So they can serve as the centerpiece of the manufacturing process. And once these crucial steps are done, we could upscale our production. And one option could be, for instance, to transfer the cells from the Prodigy into an external expansion system. And then once we have achieved our desired cell numbers, we can bring the cells back into the CliniMACS Prodigy for final formulation. This option is already enabled using a customized application program. Alternatively, a second option could be also a good alternative, and this would involve the generation of a small NK cell batch with the CliniMACS Prodigy, the freeze of aliquots of this batch, and then thaw them for later large-scale expansion in, for instance, bioreactors, and then freeze again the cells after this larger batch production to have them ready for off-the-shelf for infusion into patients. In the next part of the webinar, we are going to zoom into the expansion step and learn how we can enable a robust ex vivo expansion of our NK cells. Ex vivo expansion is a key step in the manufacturing of most NK cell-based therapies, in particular for allogenic and off-the-shelf applications. In order to develop a good expansion protocol, there are several things that we need to keep in mind. as for instance, the type of NK cells that we want to expand. Are they conventional NK cells, or do they have specific phenotypes like adaptive or memory-like? This is important because depending on the NK cells we want to expand, we need to consider their particular stimuli needed for their proliferation in order to have an efficient proliferation of the cells. Another thing that we could keep in mind is the total number of NK cells that we need to achieve for our clinical applications, and also whether we may or may not need to consider the use of feeder cells in our expansion protocol. So these are just a few considerations, but one common thing for every NK cell expansion process is the need of a good culture medium to support the expansion of the cells. NK MACS medium can be this supporting medium for your NK cells. It is a xeno-free formula that enables the preferential growth of either isolated NK cells or starting from PBMCs with minimal growth of other unwanted cells. NK MACS ultimately provides robust expansion of NK cells even under feeder-free conditions, while preserving their effector functions. NK MACS is available as both the RUO and MACS GMP to ease the translation of your preclinical expansion protocol into GMP grade. And this medium has been already used in several published studies. NK MACS GMP medium is the key for the expansion of NK cells using the CliniMACS Prodigy NK cell transduction system, as we have just seen. And here I just wanted to have a few words for our expansion protocol that basically consists on a feeder cell-free culture conditions with NK MACS GMP as the basis, and supplementation with human AV serum and the cytokines IL-2 and IL-15. But as I mentioned, NK MACS has been used in other different culture setups and multiple studies, and I would like to show a few examples of some published studies. In this particular one, performed by Close and colleagues, they compared NK MACS with other commercial media to check the efficiency of NK cell expansion. And as you see in the graphs below, the arrow pointing to the darker column represents the NK cell expansion using NK MACS and IL-2. In this case, there was only IL-2 supplementation, and as you see, it was the conditions that enable the highest full expansion of NK cells, with also the lowest contamination of CD3 positive cells. But not only the cells expanded much higher and with lower contamination, they were also nicely functional, as shown in this figure, where they tested the cytotoxicity of the cells against the K562 cell line. In this other study, this group evaluated NK MACS as well as other medium to expand, in this case, cryopreserved NK cells. And they performed the study in the following way. On the first phase, they tested the different medium for 2 weeks of expansion, and then they selected 2 media for the final phase, where they expanded the cells 2 additional weeks, and they tested different doses of IL-2 in the culture. So overall, they expanded the cells for up to 28 days, and this is what they observed. After 28 days, cells that have been cultured in NK MACS with 1000 units per ml of IL-2 were the ones that achieved the highest cumulative expansion fold. And also there was very low contamination of other cells, meaning that the cultures were highly pure in content of NK cells. In this case was comparable to both media. And not only the cells also expanded nicely, but they also preserved their natural cytotoxicity after expansion in NK MACS, as we see in these figures of a killing assay. The last dataset I would like to show you corresponds to the work done by Indapta Therapeutics that kindly shared this data with us, where they compared the use of NK MACS GMP Medium with another GMP commercially available medium on their expansion setup. And with Indapta, we have put together this data in an application note that you can access if you want to learn more details. So in the next slides, I will just show a summary of the most important findings. Indapta Therapeutics works with G-NK cells that have a particular phenotype and enhanced effector functions. So they are using a proprietary feeder cell line and cytokine cocktail to expand these cells, but had to decide on a medium to support their growth. So what they did was to compare NK MACS GMP medium with another commercially available medium. And as you see in the graph on the right side of the slide, they observed that the G-NK cells expanded significantly higher in NK MACS compared to the other medium. But these cells not only expanded in much higher expansion folds, in NK MACS compared to the other medium, but they also showed enhanced ADCC responses against the different myeloma cell lines when they were co-cultured with two different monoclonal antibodies to trigger this ADCC response. And in addition, they also preserved their potential for cytokine secretion. Again, if you would like to know more details, I really invite you to have a look at these application notes that we have available on the website. Now we are coming towards the end of the webinar, and we are going to learn about flow cytometry iPSC-QC solutions for NK cell therapy manufacturing. We have seen that manufacturing of engineered NK cells involves many steps, and that it is important to define reliable assays for in-process and quality control, the so-called iPSC-QC. In the boxes in this slide, you see examples of different assays that can be performed for IPC and QC of an NK cell manufacturing process. The ones marked in green can be actually performed by flow cytometry. But if we look beyond the manufacturing part, flow cytometry assays can also be used for donor selection or patient monitoring. However, for the purpose of this webinar, I will just focus on the iPSC-QC part and discuss which instruments and reagents can help us to standardize these assays in order to get reproducible and reliable results, such are the max quant analyzers, the use of express modes with these instruments, or the reaffinity antibodies to design our panels, as well as the CAR detection reagent. In order to perform our flow cytometry iPSC-QC assays, the first thing we need is actually a flow cytometer. And here the max quant analyzers can be a perfect fit because they really make flow cytometry easy. These instruments, they are really easy to use and they provide multiple automated features such as the calibration, compensation, and auto-labeling. They have variable formats for automated sample acquisition, starting from single vial tube holders, if we only want to analyze a few samples, up to holders that can carry 96 well plates for acquisition. This software, they can be upgraded using express modes in order to standardize our data analysis, as we will see in the next slide. But the software can also be upgraded with the 21 CFR Part 11 software module in order to enable compliance for electronic records and signatures. And besides that, we can also benefit from the Smart Gain technology to ensure consistent and reliable results when manufacturing at different sites in particular. The express modes that I have just mentioned, they enable an upgrade in the level of standardization of our flow cytometric analysis. So express modes, they are flow cytometry software tools that they have been specifically designed to be used with the software of the MACS Quant analyzers for an optimized and automated measurement and analysis. These software tools, they can really simplify flow cytometry assays via predefined experiment and acquisition and analysis settings for reproducible results. And they do so by applying an automated gating that will be automatically adjusted for every data file individually to achieve optimal results. So basically, this is a nice way to standardize our instrument setup, the sample measurement, and data analysis, which is just at the push of a button. In particular, to facilitate the iPSC-QC of NK cell therapy manufacturing, we have developed the NK cell express mode package that includes several panels that are the most common panels used in iPSC-QC of such therapies, and they are available for the MACS Quant Analyzer 10 and MACS Quant Analyzer 16. This package basically includes the essential IPC and QC assays that are performed, which involve the NK cell count, analysis of NK cell composition and transduction, and also it includes NK KIR donor selection. In regards of transduction efficiency analysis, we can also benefit from the use of CAR detection reagents. The CAR detection reagents are biotinylated antigen-based fusion proteins that basically contain the part of the target molecule that is recognized by a particular CAR and the specifically mutated human IgG1 Fc part of an antibody. So meaning that in this way we prevent the binding to Fc receptors and therefore there is no need for FcR blocking. We provide different CAR detection reagents so far with the target molecules CD19, BCMA, CD22 and CD33. And as an example, in the dot plots on the bottom right, you can see how they can perform in terms of transduction efficiency of CAR NK cells. So in this case, the NK cells were transduced to express CD19 CAR. And as you see, by using the CD19 CAR detection reagent, we were nicely able to detect the presence of the CAR on the NK cells. With this, we have come to the end of the webinar, and before we move on into the Q&A session, I just would like to show quickly 5 main take-home messages that I really would like you to take with you after this webinar. So first of all, I really want to raise a point on the fact of thinking translational during your preclinical developments to ensure a smooth transition into clinical grade manufacturing of your protocol. We have learned in the webinar that the CliniMACS Prodigy NK cell transduction enables the automated and standardized clinical grade manufacturing of gene-engineered NK cells using, in this case, viral transduction in the CliniMACS Prodigy platform. This platform can be combined with the CliniMACS electroporator to enable complex gene engineering of NK cells. Furthermore, NK MACS Medium, both as RUO and GMP grade, strongly supports the ex vivo expansion of NK cells and preserves their functionality after expansion. And finally, the MACS Quant analyzers, together the NK cell express mode package, the reaffinity antibodies, and CAR detection reagents, simplify flow cytometry-based assays for the iPSC-QC during NK cell therapy manufacturing. And with this, I thank you for your attention